Hello dear students, welcome you all to the Power Hour session. Myself Rani sir, and today we will be discussing about the chemistry chapter of biomolecules. So this is a very important chapter dear students because biomolecules is also a chapter in zoology, and over there also you are studying about the biomolecules. The only difference is that in chemistry you will be having the reactions of carbohydrates, you will be studying about the zwitter ion and the pH at which zwitter ion formation takes place. All this will be numerical based. So you are having some numerical type approach in case of this zwitter ion formation, where we are having calculation of pH, where the pH at which zwitter ion is formed is called the isoelectric point. So this is a very interesting chapter dear students, and many times a good question is asked from this chapter. So let us continue. So let us see biomolecules. Let us first define them. So what do you think? What are the biomolecules? So as you can see, there are many compounds which are responsible for living organisms to live. Like human beings, we are having carbohydrates, proteins, vitamins, and many more biomolecules which are responsible for the vital processes which are going inside our body. They play a very important role, I can say a vital role in human development and the growth of living organisms. So all these biomolecules, they are nothing but they are organic compounds which are responsible for the growth and well-being of living organisms. And what are they? What are the examples of these biomolecules? So let us see, we will be talking about carbohydrates over here. Carbohydrates, proteins, they are the very important components of our food. As we can see, the proteins and carbohydrates, they are the energy source for us, okay? And we will also be talking about vitamins, enzymes. All these are our biomolecules. So one by one we will be discussing about them. Let us see the first one, that is carbohydrates. We will be first talking about carbohydrates. So what are the carbohydrates? How you can define them? Dear students, let us see, let us define the carbohydrates. So like their name, carbohydrate, initially it was thought that they are nothing but hydrides of carbon. Initially they were thought as hydrides of carbon. Hydrides of carbon. So they were named carbohydrates, and the general formula CxH2O times. Okay? But when the study was made, a very important interesting thing happened. They saw that there are many molecules such as acetic acid, CH3COH. You can see C2H2O 2 times. It is following the general formula of carbohydrate. This is nothing but acetic acid. It is not a carbohydrate, but it follows. So it is not the case that everyone following this formula CxH2O is a carbohydrate. You can see this is acetic acid. It is following the formula, but it is not a carbohydrate. Similarly, you can see there are many carbohydrates such as rhamnose. I am talking about rhamnose. it is having the formula C6H12O5. So you can see over here, it is not following the general formula, but it is a carbohydrate. So from this you came to conclusion that initially they thought that they are hydrides of carbon, and they thought that the general formula will be CXH2Y times, but it is not the case always. Most of them may follow the formula CXH2Y times, the general formula, but there are many which are carbohydrates but they still do not follow the formula, and there are many which follow the general formula but they are not the carbohydrates. So how we should define these carbohydrates? So the best way is, we can say simply they are polyhydroxy aldehydes or polyhydroxy ketones. You can say they are polyhydroxy aldehydes or polyhydroxy ketones. And in short, we can say them as aldos because they are having aldehyde, and we can call them as ketones because they are having ketone as the principal functional group. So you can say either carbohydrates are polyhydroxyaldehydes or polyhydroxy ketones. Okay? Further, we can classify these carbohydrates. Many times they have seen this, that there are many carbohydrates which are sweet, like I am talking about lactose. Lactose is much sweeter. It is found in milk sugar. And in Greek, we call sweet sugar as saccharon. So from this saccharon, the Greek word, we started calling carbohydrates as saccharides. So we are calling them as saccharides because of the Greek word saccharon, which means sweet sugar. So let us see now, classify this carbohydrates which we are calling as them as saccharides. So let us classification of carbohydrates. So carbohydrates are classified into three types: monosaccharides, oligosaccharides, and polysaccharides. You can see we are having three types of carbohydrates: monosaccharides, oligosaccharides, and polysaccharides. As I already told you, we are also calling them as saccharides because of the Greek word saccharon. So what are the monosaccharides? So monosaccharides basically carbohydrates which cannot be further divided or broken down upon hydrolysis. They are the smallest carbohydrates, monosaccharides. On further hydrolysis of them, you cannot get a further breakdown over here. So all these example for this are glucose, then mannose, allose, and galactose. All these are examples of monosaccharides. Let us see what are oligosaccharides. Dear students, oligosaccharides are those carbohydrates which on hydrolysis give 2 to 10 monosaccharide units. What are oligosaccharides? Carbohydrates which on hydrolysis give 2 to 10 units of monosaccharides. Okay? Yes or no? So what are the example? What are the example? Mainly we will be talking about disaccharides. Disaccharides over here. And all the disaccharides, of course, they are the sub part of this oligosaccharides. Disaccharides like sucrose, or you can say maltose, lactose, all these are carbohydrates, oligosaccharides, and further we can say disaccharides. Now we are talking about polysaccharides. How you will define the polysaccharides? See, if you are getting more than 10 monosaccharide units on hydrolysis, we call them as polysaccharides. Or you can say there are many units of monosaccharides which are obtained by hydrolysis of these polysaccharides. So the carbohydrates giving more than 10 units of monosaccharides on hydrolysis are called polysaccharides. Okay, and what will be the examples? So in this you can see we are having example like starch, cellulose, glycogen. In this starch and cellulose, they are obtained from plants, and this is glycogen is found in animals. So it is also called, this glycogen is also called as animal storage carbohydrate. Okay. So this is the classification of the carbohydrates. Okay. In this monosaccharides also we can further classify. In case of monosaccharides further classified okay. So let us see. Here also we can have the nomenclature like we had in the case of organic chemistry IUPAC nomenclature. Here in case of monosaccharides also we can have that. Monosaccharides, see, as we can say, if the functional group, principal functional group is aldehyde, then this monosaccharides we call them as aldose. And if they are having functional group as ketone, we call them as ketose. This you are having the example of glucose. Everybody knows example of glucose. Okay. And in ketone we are having the example of fructose. Also we are having its epimers like tagatose, psychose, here also along with glucose its epimers like mannose, allose, galactose. So we can classify this aldose and ketose, and we can have at least 3 carbon required for carbohydrate. Okay. So we will be starting from 3 carbon, number of carbons, and then after that we are having suffix, okay, suffix in case of if it is aldose, in case of aldose what will be the prefix, and if in case of ketose what will be the prefix. So let us see one by one. Number of carbon is 3, you will have the suffix triose, and you will be having, if it is aldehyde, aldose, you will be having prefix as aldo, so you will call it as aldotriose. And if you are having ketose, then you will call it as ketotriose. Okay? If you are having number of carbon 4, you will call it as tetrose as the suffix. So prefix you will be having aldose, then aldo as the prefix, aldotetrose. And if you are having ketose, then it will be keto as the prefix, ketotetrose. Similarly, if you are having 5 number of carbon, the suffix will be pentose. Remember pentose sugars? Yes or no? So this will be aldopentose. This will be ketopentose. And if you are having 6 number of carbon, we call it as hexose. Remember all this I have given over here. All these are nothing but hexose, and these are also hexose. The only difference is these are aldohexose and these are ketohexose. So same thing I am writing over here. Aldohexose and ketohexose. Ketohexose. And in NCERT they have given till 7. So till 7 you have to see them. This will be heptose as the suffix, and so they will be called, if aldehyde has the functional group, aldoheptose, and in case of ketose, ketone as the functional group, ketoheptose. So this was all about the classification of these monosaccharides and how to name them, okay? And we will be only talking about mostly this hexose, like the aldohexoses and the ketohexoses, okay? So this was the interesting classification. Let us move further. Now we are talking about the configuration. We call it as D and L configuration. So what this basically means? What is D and L configuration? See, many students think that this capital D and capital L has something to do with dextro or levo, but that is not the case. For dextro we are using small d, and for levo we are using small l. So this capital D and capital L, they are having this configuration on the basis of configuration at last chiral carbon. Last chiral carbon will decide whether it is capital D or capital L. Okay? And this is, I am talking about in monosaccharides. In monosaccharides, what you have to do? You have to see the configuration of the last chiral carbon. If the last chiral carbon, for example, in case of glucose, for example, I am writing down the structure. Suppose I am writing down the Fischer projection over here. So you can see this is nothing but C6H12O6, and you can see here is the last chiral carbon, and OH is on the right side. So basically, if OH is on the right side, then you have to use the capital D configuration. So we will call it as which glucose? This will be D glucose. This will be D glucose. Okay? And mirror image of this. You can see mirror image of this. Can you see this last chiral carbon? Now this last chiral carbon is having this OH on left side. Okay? So if OH is on left side, I am talking about this OH. Either you have to see the OH group, and if OH group is not there, you have to see the NH2 group. Okay, if OH group is on the left side, this is nothing but L configuration. So this is nothing but, this is L glucose. So I told you about how to see the capital D and capital L configuration over here. It was very simple, because if you see the OH group on the right side, you have to say it as capital D. If you see OH group on the left side, you have to say it as capital L. If it is same in the case of NH2 also, if it is on the right hand side, then capital D, and if it is on the left hand side, then capital L. As simple as that. So I think you understood about the capital D and capital L configuration. So let us see. Now we will be talking about epimers. We are talking about the epimers. What are epimers? Anyone? See, dear students, epimers are nothing but they are having same molecular formula, and there will be only change in configuration at one chiral carbon over here. Only OH will be having different configuration at only one chiral carbon. So such carbohydrates, you can say, or you can say monosaccharides, such monosaccharides are called as epimers. They are epimeric to each other. Okay? So I am writing down the definition. Change in configuration at only one chiral carbon. Such monosaccharides with same molecular formula, are called epimers. Okay? They are called epimers. So let us see what are the epimers of D glucose. Now only we saw the Fisher projection over here for D glucose. This was here OHH, here HOH, OHH, OHH, CH2OH. This is nothing but D glucose. Now we are having change in configuration. Suppose I am having numbering over here, because this is functional group, numbering start from this side 1, 2, 3, 4, 5, 6. So I am having change in configuration. Suppose at the second carbon, change in configuration at second carbon. So if we are having change in configuration at this second carbon, what will happen? You can see we are getting this You can see over here, we are getting this, and it is having change in configuration at this second position. So this is called D-mannose. The name of this is D-mannose. I am writing over here. This is nothing but D-mannose. So what is the relation between D-glucose and D-mannose? They are epimers. They are epimers. Similarly, I am having change in configuration at third carbon. I am having change in configuration at third carbon. So what will happen? Let us have the change in configuration at third carbon. C double bond OH, OH, H. This is the third carbon. So this is OH, H, OH, H, OH, H, and CH2OH. You can see we are having change in position, or you can say configuration of the OH or the third position. At the third position we are having change in the configuration of OH. So this is also epimer, and the name of this epimer is D-allose. So what we can say, D-allose and D-glucose are epimers. Now we are having change in configuration, suppose at the fourth chiral carbon. Change in configuration at fourth chiral carbon. So what will happen? What will happen? Fourth carbon we are having this change over here. This fourth carbon we are having change of the configuration. You will be getting C double bond O, H, OH, H, HOH, and then here onwards this is the change, HOH, then OHH, CH2OH. You can see we had the changing configuration at which chiral carbon? We can say at which carbon? 1, 2, 3, 4, fourth carbon, okay? And the name of this is D-galactose. D-galactose. As you can see, they are the epimers of D-glucose over here, okay? Similarly, like D-glucose, we had this epimers, we have epimers for D-fructose as well. So let us see epimers of D-fructose. D-fructose epimers, beta. Let us see them. So I am writing down the structure of this D-fructose. Everybody knows it is a ketose. So this is nothing but D-fructose. Because you can see at the last chiral carbon, you can see OH is on the right side. This is D-fructose. And now we have to draw the epimers of it. Everybody knows how epimers will be drawn. We will be having change in configuration at any one of the chiral carbon. So 1, 2, 3, 4, 5, 6. This is the numbering. And let us have change in position or change in configuration at the third carbon. Change in configuration at third carbon. Change in configuration at third carbon. You can see I am having change in configuration at third carbon. You can see I am having changing configuration at the third carbon. And what is the name of this? This is nothing but D-cyclose, which is a epimer of D-fructose. Now we are having changing configuration at third. Now we will be having at fourth. Change in configuration at fourth carbon. So let us have the changing configuration at this fourth carbon. What will happen? We are getting CH2OH, C double bond O, HOH, HOH, OHH and CH2OH. You can see we had the change in configuration at the fourth carbon, and this is nothing but D-tagatose. So you can say D-tagatose and D-cyclose, they are the epimers of D-fructose. So this was all about the epimers. As you can see, I told you the epimers about D-glucose and the D-fructose. So let us see about anomers. What are the anomers? Let us see the anomers, okay? So anomers, basically what will be the definition of anomer? See, we are having this straight chain structure of the D-glucose or D-fructose, as you can see. And now what will happen? This one of the, or you can say last chiral carbon having the OH group on it will attack as a nucleophile on the aldehyde or ketone, and then we will be having a cyclic structure, okay? And while this attack, you can see we will having a formation of a new chiral center which can have both the configurations. So we can have the anomers over here. These are the anomers where this last chiral carbon OH will attack the aldehyde or ketone group, and there will be formation of a cyclic molecule. It will be having two options, either alpha or beta. So these anomers are these alpha and beta forms, okay? So let us see the definition over here. What is the definition of anomers? The OH group on last chiral carbon of monosaccharides attacks as a nucleophile on aldehyde or ketone group, okay? And a new chiral center, formation take place. See, dear students, if a new chiral center is forming, there are two possibilities. So we will be having two anomer formation take place. And we will be denoting it by alpha and beta, okay? So from D-glucose, you will be getting the anomers alpha D-glucose plus beta D-glucose. Similarly, from D-fructose, you will be getting the anomers alpha D-fructose, beta D-fructose. If you draw the Haworth projection of this, so simply if you draw the Haworth projection over here, I am telling you a very important point, that in case of this alpha D-glucose, you will be having, in case of this alpha D-glucose, you will be having a structure like a pyran. So you are having this suffix pyranose over here, okay? And in case of this fructose, we are having a structure like furan. in case of this. So in Howard projection, we are writing the suffix as furanose over here, furanose. So this is a very important point, that in case of Howard projection, we have to add the suffix. We have to add the suffix over here. In case of this D-glucose, we are having this pyran structure formation, so we will be having pyranose as a suffix, and in case of this D-fructose, we are having furan structure form, so we will be having suffix added as furanose. Okay, so these are nothing but the anomers that we got from the D-glucose and D-fructose over here. Okay, so this was all about the monosaccharides. I also told you about disaccharides. See, disaccharides basically they will be formed from two monosaccharides. Like the fructose and glucose. You can say alpha D glucose and beta D fructose. Alpha D glucose and beta D fructose. These two will form together. By removing water, you will get sucrose. Okay? And this sucrose is, if you see, it is generally optically active, and positive means it is a dextro over here. A very interesting fact I am telling you, this is dextro, but if you hydrolyze it, you will get this too, and this is having higher value of negative angle of rotation. So what happens if you hydrolyze this sucrose, you will get the mixture of these two, and it will be overall levorotatory. So by having this sucrose hydrolysis, if you are having this hydrolysis, you will get from positive to negative, and that's why sucrose is also called invert sugar. We also call it as invert sugar. Okay, so this was the first disaccharide, sucrose. We can also have maltose formation over here. Okay, like alpha D glucose plus alpha D glucose. Then we are removing the water to get the glycosidic linkage, and you will get the maltose over here. Okay, next we can have lactose formation also. Beta D glucose and beta D galactose. And by removing the water, we will be having the formation of lactose over here. In this C1C2, C1C4, C1C4. So that we have to remember over here. Okay. So let us see the questions. We already covered the monosaccharide part. Just a few things I am adding over here. This sucrose is giving negative test for Fehling, Benedict. So it is a non-reducing, whereas these two are reducing. So let us see what are reducing and non-reducing. So reducing sugar are those which will give test for tolerance test. You can say it will give positive test for, or you can say they will give positive tolerance test or Fehling, Benedict test. They will give positive Fehling or Benedict test. Okay, these are the reducing sugar. Whereas non-reducing sugar, they will give negative tolerance test. Everybody knows tolerance test, AgNO3 plus NH4OH, and negative Fehling or Benedict test. So what are the examples of this? So all monosaccharides gives this reducing sugar and disaccharides except sucrose, and over here we are having sucrose and all polysaccharides. Everybody knows polysaccharides. I give the example: starch, cellulose, glycogen. All this will be having non-reducing sugar. They will be having negative test for tolerance test and Benedict and Fehling test. Okay, so let us see. Now we are ready. Now let us see the questions over here. So this is the question they are asking. Match the column. In column 1 we are having carbohydrates, and they are asking us what will be the source from where we will get this carbohydrate. So sucrose, everybody knows cane sugar, from there we get the sucrose, which we are having in our house. That sugar is nothing but sucrose only, beta. Okay, so A will be having this S. Lactose, I already discussed milk sugar. Milk is having this lactose. Glycogen, as we already discussed, aliment storage, as we discussed over here, this will be P. And starch found in cereal, roots and tubers, so this will be Q. So our correct answer will be this B option over here. Okay, so let us see the next question. Glucose, see they are having this reaction of glucose with HI. See, this is a very good reducing agent. Generally we use red phosphorus plus HI, but in case of this glucose, only HI is sufficient. What will happen? It will completely reduce. This glucose, and we will be getting N-hexane over here. N-hexane. Okay. So we are having for glucose on reaction with HI heat, we will be getting N-hexane. So for A, we are getting Q over here. Now we are having glucose. Glucose reacted with bromine water. So this is a very weak oxidizing agent. You can say mild oxidizing agent. It will be oxidizing this glucose to gluconic acid. Everybody knows the structure of glucose. I told you CHO on the heading, aldehyde group. That CHO will be converted into carboxylic by this bromine water, and that name of that acid is gluconic acid. So we are getting for this BSR. Now we are reacting with glucose. Glucose is reacted with nitric acid over here. So this nitric acid is a very good oxidizing agent. Both the CHO will be oxidized to COH, as well as the last CH2OH will be also oxidized to COH. Above also COH, and the last carbon also COH, and that is our saccharic acid. So glucose will give saccharic acid on reacting with nitric acid, and then we are reacting with glucose, and this is nothing but 3PH H plus NH2. So this is nothing but, you already know, oxazone formation will be taking place over here. So this D will be having P. So the answer will be AQ, BR, CS, DP. So D option will be our correct answer. So let us see the next question. Maltose. Now only I told you maltose is prepared from 2-alpha D glucose. Lactose. Lactose is prepared from beta D galactose and beta D glucose. Cellulose is prepared from beta D glucose. And sucrose is prepared from alpha D glucose and beta D fructose. So what will be the answer? For A, we will be having Q. for B we will be having R, for C we will be having P, and for D we will be having S. So B option will be the answer. So next, let us see. So these are our cyclic structure as I told you. That's why I told you we have to write the suffix in case of glucose as glucopyranose, in case of fructose as fructofuranose. So these are the Haworth projection over here given. So I can see it is a 6 carbon one. So it will be basically a hexose, and we are having two options, either alpha D mannose or beta or alpha D glucose. I can see over here this HOH. So definitely this A will be our R, that is alpha D glucose. Over here you can see second position. As I told you, as I taught you epimers, at the second position I told you that alpha D glucose, the C2 position epimer is nothing but mannose. So you can see that help we are getting over here. In this, the only difference is at the C2 position 1 and 2, you can see the second position we are having change in configuration. So definitely if this is D glucose, this is D mannose. So this is having P. Okay. So let us see the options. We are getting ASR, ASR and BHP. ASR and BHP. Either the answer will be this one or this one. So let us continue. C, for C you can see C, this is nothing but, as I can see, this one is having OH upward, and so I can see that this is nothing but a beta one. So beta, C will be having Q, and this is nothing but alpha, change in configuration at this. This is nothing but the anomeric carbon beta. As I told you, anomer. So these two are anomers. This is nothing but beta D fructose, and this is nothing but alpha D fructose. The answer will be, so the answer will be over here. This is P option. Okay. Next, which of the following are non-reducing sugar? Now only I have discussed that all polymers, polysaccharides, cellulose, starch, they are nothing but non-reducing, as well as this disaccharide, sucrose is also non-reducing. So our answer will be A, B and D. A, B and D, the C option will be the correct answer. Okay. So next, given below are the two statements. One is labeled as assertion and the other as is labeled as reason. So let us see this assertion reasons. Assertion, glucose gets oxidized to 6 carbon carboxylic gluconic on reaction with mild oxidizing agent like beer to water. Now only we discuss this. This is correct statement. Correct. Assertion is a correct statement. Next, reason. This indicates the presence of a primary alcoholic OH group in glucose. See, this is not a correct reason, I think, because they should say that because of the aldehyde group, that Br2 water can oxidize the aldehyde group to carboxylic acid. So there should be a reason because of the presence of aldehyde group. So this is not a correct reason over here. So I will go with assertion is true but reason is false. Okay. Next, given below are the two statements. Statement 1, the pentacetate of glucose does not react with hydroxylamine, indicating the absence of free CHO group. Okay, this is correct statement. Second statement, despite having the aldehyde group, glucose does not give Skiff test and it does not form the hydrogen sulfide addition product with NaHSO3. Yes, this is also correct statement. See, this actually is connected with the carbonyl compound and carboxylic acid. Over there we had studied about this. So this B option is the correct one. Let us see the next one. All carbohydrates which reduce Fehling solution tolerance reagent are referred to as reducing sugars. It is a correct statement. All disaccharides, whether aldose or ketose, are reducing sugars. See, I had told you that one of the disaccharide, like the sucrose is not a reducing. So this is a incorrect statement. So statement 1 is correct, statement 2 is incorrect. C option will be the answer. Next. Now let us see the another assertion reason or statement 1, statement 2 question is there. Sucrose is levorotatory, but sucrose is dextrorotatory. This is incorrect. So directly I can see statement 1 is incorrect. Statement 2, hydrolysis of sucrose brings about a change in the sign of rotation. Yes, correct. This is the correct statement. We already discussed this, dear students. Now only we discuss this. That positive it is first dextrorotatory and after the hydrolysis, the overall it is levorotatory. So they have given, yes, the same thing they have given. So second statement is the correct statement. So answer will be, what will be the correct answer over here? Statement 1 is correct, incorrect, and statement 2 is correct. So the B option will be our answer over here. Next, given below are the two statements. Amylose is water soluble component which consists of about 15 to 20% of starch. Chemically, amylose is a long unbranched chain with 200 alpha D glucose units held at C1C4. Okay, this is the correct statement. Statement 2, amylopectin is insoluble in water and constitutes about 80 to 85% of starch. Yeah, this is also correct statement. Let us see, this is having C1C4 glycosyl linkage and branching at C1C6. Yeah, they are giving us the correct, both the statements are correct over here. So answer will be our A option. Next, statement 1, only alpha amino acids are obtained on hydrolysis of proteins. Yeah, we are having these proteins as nothing but they are the polymer of alpha amino acids. The amino acid which can be synthesized in body are known as essential. No, the one which are synthesized in body, we are calling them as non-essential. So this statement 2 is incorrect. So the answer will be statement 1 is correct and statement 2 is incorrect. D option. Next, in zwitterionic form, amino acids shows amphoteric behavior as they react with both acid and base. Yes, because see, this amino acid are having NH2 also, which is a base, and COH, which is acid. So at the zwitterionic point, this H plus from COH goes to the NH2 and we get the zwitterion. So it is a salt. Definitely they are giving the correct statement. Statement 1 is correct. Statement 2, most natural occurring amino acids have D configuration. I think they are having L configuration. In case of carbohydrates, they are correct. But they are not talking about carbohydrates. They are talking about amino acids. And in case of amino acids, most of them are having L configuration. So they have given a wrong statement too. So the answer will be statement 1 is correct, statement 2 is incorrect. The B option will be the answer. Next, so let us see, this is a biology type question, but in chemistry we are having this. In the NCERT they have given. So we have to remember vitamin A. See, vitamin A we are getting this fish liver oil. Vitamin B12 we are getting it from meat. Vitamin B6 we are getting it from yeast or milk. And vitamin D we are getting it from exposure to sunlight. So the answer will be A option. Next, this also question in NCERT they are giving this. So we have to give them priority because many times they are asking questions from NCERT. Vitamin B1. So this vitamin B1 I can see it is found in this berry. Vitamin B6 deficiency, okay, they are asking us disease. So vitamin B1 deficiency will cause beriberi disease. Vitamin B6 will cause convulsions, that is the S, and vitamin C will cause scurvy, and vitamin K deficiency will cause blood clotting delays. Okay, so the option will be ARSPQ, AR, BS, CP and DQ. So C option will be the answer over here. So this was a question where vitamins and their deficiency diseases, they were asking vitamin B1, we already know beriberi disease, vitamin B6 we are having this convulsions, vitamin C we are having scurvy, and vitamin K we are having blood clotting delays. Okay. Next question is fat soluble vitamin. So everybody know this, there are two type of vitamins, fat soluble and water soluble. Fat soluble are vitamin A, D, E, K, and water soluble are B and C. So let us see fat soluble vitamin A and D. Everybody knows this. Water soluble B and C. Yes or no? And we are having DNA. So this DNA will be having fingerprinting, and RNA will be having uracil over here. So the answer will be A, Q, B, S, C, P, and D, R. So B option will be our correct answer over here. Next, heredity. Everybody knows heredity is nothing but transmission of inherit characters. In NCERT, in chemistry also, they have given this line. So this is a very important question. Chromosomes are made up of, they are made up of nothing but, we already know they are made up of proteins. Next, nucleic acids, they are long chain polymers of nucleotides, and vitamin E deficiency will cause increased fragility of RBCs. So we are having for heredity this Q, for chromosomes made up of proteins, nucleic acid S, and vitamin E deficiency R. So A, Q, B, P, nucleic acid C, S, and D, R. So A option will be our correct answer over here. Next, assertion reason, one more we are having assertion. Amino acids are water soluble, high melting solids, and behave like salts rather than simple amines or carb. Already we discussed this, that because of this both groups, this H plus from COH will be going to the NH2, and we will be having a salt formation. And already you know that the salts, they are having high melting point because they are ionic compounds. So next reason, this behavior is due to the presence of both acidic COH and basic NH2 group in the same molecule. Yes, this is the correct reason. Only because of that this thing has happened. So we are having over here both assertion reason are true and reason is the correct explanation. Next, statement 1. Protein found in a biological system with unique three-dimensional structure and biological activity is called a native protein. Yeah, this is the correct statement. Statement 2. Except glycine, all other naturally occurring amino acids are optically active. See, glycine is the smallest amino acid. See, this is glycine. So it is not having any chiral carbon. So this is optically inactive. So they have correctly said it. Except this glycine, all this alpha amino acids are optically active. So this is also a correct statement. So the answer will be both statement 1 and 2 are correct. Okay. Next. Statement 1. Enzymes which do not catalyze the oxidation of one substrate with simultaneous reduction of another substrate are named as oxidoreductase. Why they have said this do not? Actually do it should be. So this is a incorrect, sorry, it is an incorrect statement. And statement 2. Enzymes are needed in large quantity. See, enzymes are catalyst. Why we need this catalyst in large quantity? Everybody knows that catalysts are required in less quantity. So they have given again a wrong statement over here. So as you can see, both statement 1 and statement 2, both are incorrect over here. So C option will be the answer. Next, read given statements. During denaturization, 2 degree and 3 degree structures of protein are not destroyed. See, 2 degree and 3 degree structures of protein are actually, I can see they have written are not destroyed over here. So this is actually, I can see over here, an incorrect statement. because 2 degree and 3 degree are destroyed and 1 degree remains as it is. So they have given a completely wrong statement over here, because 2 degree 3 degree gets destroyed while 1 degree remains as it is. And the statement 2 they have given 1 degree structure protein is destroyed, 1 degree remains undestroyed. So second statement, this B statement, is also incorrect one. This is also incorrect one. Both this A and B, both of them are incorrect. C, curdling of milk which is caused due to formation of lactic acid by bacteria present in milk is an example of denaturization. Yeah, this is a correct statement. Everybody knows that this, while having the temperature change, you will be having this denaturization, where the 1 degree structure will remain same, undestroyed, whereas 2 degree 3 degree will be destroyed. So this statement is correct, and they are asking us which statement are incorrect. So as we discussed, this A and B are incorrect over here, so the option correct will be B option. So that was all about this biomolecules session over here. Hope you enjoyed this session. So let us meet in the next session, which is about P block. For the chemistry I am talking, we will be having power hour session for the P block in our next power hour session. So hope you enjoyed this power hour session. Thank you dear students for joining. Let us meet in the next power hour